This network is demonstrating the formation of spikes. I will be using an analogy for baking a spike cake. So S stands for water, P stands for cake mix, I stands for sprinkles, K for eggs, and E for oil, and this is our spike cake. The wild type version of this network is as follows. S, the water, promotes P, the cake mix. P, the cake mix, then blocks I, the sprinkles, so then I, the sprinkles, cannot block K, the eggs, from promoting E, the oil, which then produces the spike cake. So if we are lacking P, the cake mix, then there will be no spike cake produced. This is because if P, the cake mix, does not block I, then I will block K, the eggs, from promoting E, the oil, which then E, the oil, bakes the spike cake. So if you have no cake mix P, that means no spike cake. If we are lacking I, the sprinkles, then we are still able to make spike cake because sprinkles is not an imperative ingredient. Without I, we are still able to promote E because K is no longer being blocked by I. If we are lacking I, sprinkles, and E, oil, then we will not be able to make spike cake. If we do not have I, then we can still make the cake as demonstrated above, but if we do not have E, oil, we will not be able to make the spike cake because E's only job is to bake the spike cake, so without E, we can't bake the spike cake. So if we lack S, water, and K, eggs, then we cannot make spike cake. This is because these are imperative ingredients. S promotes P, which blocks I from blocking K, which promotes E, which then produces spike cake. So without the water, we allow P to not block I from blocking K. So if we lack K, eggs, this is an imperative ingredient, so the cake will not be made. K, eggs' job is to promote E, oil, which then makes the cake. Without K, E will not be making the spike cake. So without K, eggs, the cake will not be made. Methylation, as demonstrated with the dragon as the histone and the band as the DNA. Methylation is when DNA methyltransferase turns genes off and silences them. So even though the genotypes have not changed, the expression of that gene is turned off. So DNA methyltransferase is an enzyme that transfers a methyl group onto the DNA, which then turns the genes off. These sites that are methylated are called CpG sequences. These CpG sequences, cytosine, phosphate, guanine, attract deacetylases, which then remove the acetyl group. So they make DNA coiling around the histone tight, which means more competition, and the outcome of this increased methylation is decreased transcription of the DNA. So for our example, a methyl group attaches to the P, our cake mix, which turns the spike cake mix genes off, so the spike cake was not made even though we technically had the genes for it because of this methylation.